Namaskar, Jai Hind, and good morning to everyone. Welcome to day 2 of PDNS 2021. After a fascinating first day, we are eagerly looking forward to an equally fascinating second day with some very interesting sessions and presentations coming up. The subject or the sub-theme of this morning's session is dealing with future challenges and the role of science and technology in that aspect, in that context. We have two outstanding speakers and another outstanding person as the chairperson. So we are very fortunate to be able to get them to agree to participate in this. And without further ado, I'm going to hand over the session to Dr. Sangeeta Kale. But before that, a few words by way of introduction would be in order. Dr. Sangeeta Kale is a professor at the Defense Institute of Advanced Technology, DIAT, as sometimes referred to, which is at Khadakwasla, close to Pune. She did a master's in electronic science and doctoral studies from the University of Pune, and then postdoctoral studies from the University of Maryland in the United States from 2000 to 2002. She's been working at the DIAT as professor and dean of that university, and is also a visiting scientist at International Center for Theoretical Physics at Trieste in Italy. Actually, she has such impeccable credentials. It'll take me a long time to go through all of them. So I'm just going to highlight just a few more before handing over the session to her, because we are very keen to listen to what she and the other two speakers have to say. Her work includes nanotechnology and its application in the domain of CBW sensors, electromagnetic shielding of radars, optical sensors for field detection, metamaterials for smart detection, and drug delivery using nanomaterials. All of these are so relevant to in the field of the defense services. She has successfully executed projects which are funded nationally from the Department of Science and Technology, ISRO, DRDO, Department of Atomic Energy, and you name it, several others. At this point, I must say that after all this, she still finds time to host a very popular show, which PIC has every month, called PIC Adda, where we listen to people from various fields with various backgrounds on various interesting topics. And she hosts that show for the PIC. So I don't know how you manage your time to do all this, but over to you, Dr. Kale.
Thank you, sir. Thank you, General Patkar, for a very warm introduction. I also warmly welcome Dr. R.A. Mishra sir, Dr. Vijay Kelkar, Mr. Abhay Vaidya to this session. At the outset, let me express my sincere gratitude towards Mr. Madhukar Kotwal, who was actually supposed to chair this session. However, due to some unavoidable situations at home, he was not able to do so, and he has requested me to conduct this session. However, he had done huge survey on this topic and has been instrumental in identifying excellent speakers for today's session. I thank him for this, and good morning and welcome to this session, which is dealing with future challenges, role of science and technology. As we all know, role of science and technology is meeting future challenges in disaster risk reduction. This is a vast topic. The task of managing disaster risk and disaster events is heavily dependent upon scientific knowledge and evidence-based techniques. The application of science and technology can substantially reduce losses of lives and the property. For example, if we think of floods, then early evacuation warning systems would be highly useful, and they are useful. For situations like hurricane, better constructions, particular through checking processes of engineering, would be highly useful. Earthquake, for example, soil liquefaction is a process which can be used, and this all requires good amount of science and technology. Therefore, at a broad level, natural disasters can be divided generally into following broad categories like geophysical, for example, earthquakes, hydrological, for example, floods, meteorological, such as cyclones and tornadoes, climatological, such as droughts and wildfires, biological, such as pandemics, insect infections, and so on, extraterrestrial, such as solar flares and space weather. Extent of annual damage recorded has been ranging from around $150 billion to $400 billion in the last decade before the COVID times. The loss of human lives, although steadily decreasing, it has amounted to around 60,000 per year on an average to over last decade pre-COVID again, which is exceeding 3 lakhs in the past decade. So the question is that do policymakers really endorse the use of science and technology for the disaster risk reduction? We'll try and understand in this particular session. Secondly, and most importantly for this session, current pandemic times have been classic example of disaster, natural and otherwise. This topic is important not only for the disaster management through development of science and technology, but also to the implementers or policymakers. I've gone through a recent OECD report which has been published in June 2021, which states that the COVID-19 crisis could have substantial effects on the future of science and technology, which we will try to understand in today's session. Such factors include an unequal effects of crisis on research and development, R&D, across the sectors, and accelerated adoption of digital tools and techniques, and changes in the openness, inclusiveness, and agility of research and innovation ecosystems. The crisis could also spur experimentation with new tools, policy approaches, and governance models. Few of them could be, for example, ensuring sustained funding of R&D by universities, public research institutions, and firms of COVID-19 crisis, strengthening and supporting the innovative business most affected by this crisis, particularly smaller firms to safeguard competitive markets. Adoption of systematic and collaborative approach to science and technology and innovation policies could be one of them. And continue to monitor and explore the critical uncertainties which are generated by pandemic and its aftermath, including plausible future disruptive changes at systemic level. Moreover, developments in disruptive technologies such as artificial intelligence, Internet of Things, big data, and innovations in the areas of robotics, drone technologies are transforming methods deployed for disaster risk reduction. Rapid advancement in computing capability such as quantum computers and in genome engineering with tools like CRISPR-Cas, while providing scope for massive improvement, they simultaneously open huge avenues, both useful and otherwise. Had it not been for incredible advance in biological sciences, especially in the field of genetics and biotechnology, one can only imagine how many more million would have been adversely impacted in this pandemic. Now, since this is a vast topic and since we have time constraint, it will be impossible to do justice on this broad area which I just spoke about. Therefore, through science and technology, though it will continue to play a crucial role, this session would primarily focus on the following two areas. One is the biotherapeutics in pandemic times, and in these times, what are the lessons that we learn for self-reliance and better surveillance? I have excellent panelists for today, Dr. Sanjay Singh and Dr. Rakesh Mishra. I will first introduce you to the speakers and then hand over to the panelist members. Dr. Sanjay Singh is a Chief Executive Officer of Gennova Biopharmaceuticals Limited in Pune, India, a biotechnology company which is dedicated to development and commercialization of safe and affordable biotherapeutics. As a founder CEO since 2006 at Gennova, Dr. Singh focused on innovations in biomanufacturing technologies that culminated into commercialization of 7 life-saving biotherapeutics in cardiovascular, neurology, nephrology, and oncology. His resume is very, very rich and very wide. I will just cover some major parts of it. Under him, Gennova has directed their research to cater to vaccine development of neglected diseases, in particular malaria, HPV, TB, in partnership with various national and global organizations, including NIH, US FDA, and so on. Dr. Singh is currently a member of various important committees at national level and international level, of which one is Indo-US Vaccine Action Program. Contributing towards national interest, Dr. Singh has contributed a lot for DBT, DST, CSCO, Ministry of Health and Family Welfare, and so on and so forth. Dr. Singh holds a PhD degree in biochemistry from Central Drug Research Institute, Lucknow, India. Before joining Gennova, he headed antigen research section at Malaria Vaccine Development Unit of National Institute of Allergy and Infectious Diseases, NIH, USA. Our second speaker is Dr. Rakesh Mishra. Dr. Rakesh Mishra is a director of Tata Institute of Genetics and Society, TIGS, India. He received his PhD from University of Allahabad, India in 1986. He did postdoctoral research at Indian Institute of Sciences, India, University of Bordeaux, France, St. Louis University, US, and University of Geneva in Switzerland. He then joined the Center for Cellular and Molecular Biology, CCMB, in Hyderabad, in India, and became director in 2016. His research interests are comparative genomics of non-coding DNA in the context of evolution of complexity, genetic basis of anterior-posterior body axis formation in animals, and role of epigenetic regulations in development. Under his leadership, the Atal Incubation Center was established at CCMB, which provides life sciences entrepreneurs the infrastructure and mentorship needed to encourage innovation. CCMB has also played a key role in genomic surveillance of the SARS-CoV virus in India during the COVID-19 pandemic. Rakesh is an elected fellow of major science academies in India and a recipient of several honors and awards, including the J.C. Bose National Fellowship. With these introductions, I welcome both the panelist members to this particular session, and I would now request the first speaker, Dr. Sanjay Singh, to take over. Over to Dr. Sanjay.
Thank you very much for your kind introduction. And I have to talk from the biotechnology side. So I have made a very small presentation, but I would like to more talk on individual slides, how to go around, and in between. Once I finish, then I think Professor Mishra will come, and then after that, I think we have 15-minute discussion. So can I share my slide? Can you see my slides,
yes,
my presentation? Is visible? Thank you. So first, thank you to Dr. Maselkar and Dr. Kale for inviting me for such an important topic. Because since this COVID has taken over in last 14, 15 months, continuously this health security comes in my mind, and from a very different aspects, not only in terms of that how we can create the cure, how we can manage these hospitals, but also what are the ways we can prevent. Because what is happening at present point, these epidemic-borne viruses were known to create these kind of threats for last 2 to 3 decades, and up to a very high extent. And when during my days at National Institute of Health, the first SARS-CoV-1 infection came, and it was, though it didn't do any comparison to this damage, but yes, it was a very, very worrisome point. Then H1N1 was there, so this was another worrisome point. And it somehow, what we are realizing, most of these pathogens which are coming, 70% in last century, they have come from the animals. So what the relationship we are having, these pathogens are not available on which we can do the research or we can understand what kind of shape or size or damage or pathophysiology they are going to come, because they are coming from the animals. So this is one of the very, very challenging part for us. The second part is that in case of COVID-19, we have seen the damages are unprecedented, and still we do not know which direction it is going, because a lot of mutations is happening, a lot of problems are happening. So whatsoever we have thought about maybe 2 years before about global health security or national health security, those parameters has totally changed. And I just want to give one example to you how I want to start the talk. So this was announced in 2020, the Global Health Index, and where the whole number were given. And the US was #1 in Global Health Index in the term of security. But if you see, in case of India was ranked around 14.6, so 57 rank out of 195. This was data which I was analyzing because this is the new kind of trend setting has happened. But if somebody is #1, if we see the next slide, sorry, if we see the #1 slide, then the damage has gone maximum to the #1. The biggest total cases and the deaths as on comparison to any other country. And then now India is the second, which was like 57 lines. So what are the factors? Means whatever we decided a couple of years ago or a decade ago, or what we're thinking, somehow they have fallen flat. So this kind of dialogues are very, very important to rethink, to re-strategize what kind of prevention we need, what kind of scanning mechanism we need, what kind of data analytics we need, what kind of health preparedness we need, what kind of tools we need to control if these kind of outbreaks happen. And I think I have made my point clear. So even 2 years before, when in 2019, this global health security data was created, and in that creation, the countries have been ranked, but when the COVID-19 came, it has affected the one who was the most prepared. It means our thought process needs to change. Now coming back, because I'm not expert in the public health, I have some interest into it, and I closely look in these areas, because when SARS-CoV-1 came, then the US decided that they will put a surveillance mechanism. And very simple part I want to tell about the surveillance mechanism, that if New York, in hospitals, roughly deaths are X number, and if more than 25% deaths happen in a week, then we have to, the surveillance system will switch on, it will go back to HHS, it will go to CDC, it will go to NIH, to say that there is some alarm system is coming, though we do not know why it is happening. And this surveillance system was developed all over the country, with majority of the places, with some points in the nationwide. But from 2000 to 2019, nothing happened to somehow they dumped that system. Not enough funding has gone into it, not enough analytics has gone into it, not enough care has been taken to that particular system. And this is the outcome of not taking care of it. So sometimes we make the policies, and with policies, when we don't see anything not happening, then we become comfortable and we drop those systems. So investment into these areas are not one-day investment. These are investment which are very long-time investment. So roughly what the economists are saying, US has lost around $10 trillion in last 2 years of economy. And hopefully next 10 to 11 trillion dollars are going to lose between now and 2025, because it has impacted everything. Similar way in our country, I think we should have grown with a very high number of GDP, but it has affected us 1/3 of our economy last year. And how many job losses has happened in terms of economics, but consider how many lives we have lost. Could we have been better prepared for it? So this health dialogue, I feel that it should focus on, because I believe two of the areas are paramount for any nation: the defense and the healthcare. And somehow in past many decades, they have been not taken on the point of innovation, what we need for next 5 years, what we need for next 10 years. So innovation in the healthcare and innovation in the area of defense is very critical. And why I'm saying these innovations are required, because our needs are different. Our needs are not exactly like needs of the West. So coming to these points, now I'm coming about what is the best way. It has been proven in last couple of centuries, the vaccines are the way. They can be preventive, they can be effective, and that is the only way when these kind of pandemic comes, we can fight it. Today, I'm just more focusing on the pandemic like COVID or any other pandemic is possibly to come. But there is one part I'm not taking, especially here, there's a part with biological warfare. If for those things we also have to prepare, and we have to prepare at a very high level, and in most of them, these vaccine technologies will make a big part. I'm a very optimistic person overall. Yes, we were not prepared, but since we moved into that direction, we have made huge advances in terms of our diagnostic technology. We became very quickly self-reliant. PPE kits, we became very quickly self-reliant. In hospital, how the treatments are having, the guidelines were established. And then finally the vaccines were having, and Government of India put so much support into it, including the Prime Minister visiting the companies, talking to the scientists. I never saw this happen before. An outcome of that we are so fortunate that we have immunized around 100 crore people. Unprecedented task for a country like India. So we have these resilience, only we have to plan it better, and we have to be preventive in this area. And this is one of the topics which today I'm going to present. So in case of vaccine technology, if we consider this, how it goes, so there are basically 4 types of technologies, platforms are available in the world. So one was whole organism. This was developed somewhere by 1796 by Professor Jenner at Oxford. And he demonstrated that you can take the whole organism, you can somehow attenuate it. And if you give this vaccine, this can take care of the disease or take care of infections, especially transmissible infections. And today also vaccines from these platforms are available, like polio, hepatitis A, rabies, influenza, yellow fever, measles, mumps, and rubella. So this is generation old technology. Then maybe 60 years back when genetic codes were deciphered and we can understand how we can make recombinant protein, around 30-35 years before, a new class of vaccine started. And this is basically the recombinant subunit vaccine. The only thing in the vaccine, one category is that for this, you have to grow the virus. So risks are again very high, just like FMD, foot and mouth disease. Wherever we make the vaccine, there is possibility there could be its own outbreak can happen from there, and it happened in the past. So you have to grow the viruses, you have to grow in very huge quantity. So there is a lot of, you have to create BSL-3 facilities. So there is a risk factor. When the science moved to the second platform, here you don't have to touch in general the virus. What you have to do, you have to, somewhere you have to sequence it, you have to get it, and then you can make a recombinant protein. But for this, you have to make the cell bank, you have to make all those kind of things. But this is a more defined culture came. And then the third generation came, it is a decade ago, which is basically the DNA-based platforms. So adenovirus or plasmid DNA vaccine. And here, one of the outbreak was taken, emergency approval was given. It was Ebola vaccine. This outbreak happened in Africa a couple of years back. And now from this platform, AstraZeneca vaccine has been approved, which is Covishield made by our peers here in Pune. And the second one is now the DNA vaccine from Zydus has been also approved. But the last platform comes, which is the mRNA platforms. And when this situation arises in front of us, what to do with this and where Gennova should go, we thought about all these, and we were already working a year or two behind on this technology, because this technology has a couple of different level of advantages. One advantage is it is pure synthetic technology. just like chemical reactions. So you don't have to grow the cells, you don't have to make the DNA in such quantity. So you can put your DNA template and add a couple of things, and this just mRNA is made. So this is a pure synthetic, and you start in the morning, by the evening, your vaccine is ready. So this was most exciting, but at that time, when we moved towards the COVID vaccine based on mRNA technology, we talked to the DBT, submitted our proposal to DBT, and DBT evaluated it, but thanks to the government that they finally gave a seed funding for us 25 crores. Because when we decided to do this, we were knowing this is going to be a Hercules task to take this vaccine for a company small like us to invest such a huge amount. So they said we will be part funding, and we started this program. Now, if I say in detail what I have said, that what are the advantages, what are the differentiation in these four. So this is vaccine one. You have to grow these viruses in high cell density. You have to infect with a live virus. You have to propagate the virus. You have to harvest the virus. You have to estimate the potency, and then you can do the formulation and you can release for the market. In case of two, the second way, this is a lot of students may be listening or people will be listening, so I just wanted to give elaboration on this. So in vaccine generation 2, where you can have the encoding gene, you have to prepare the master and working cell bank, you can make large scale cultures, and then harvest and then purify like other ways that you have to go forward for it. In case of DNA, here again, you have to only make one component, which is once you infect the cells, you can produce the DNA, and you can take out and purify the DNA and fill and finish. And in case of mRNA, another huge advantage for this is you have a DNA template, you just simply do in vitro transcription, and you can do in a tube, or you can do in a tank, or you can do in a 5,000 liter cap, and just small some chemical reactions are there. Very simple purification, because there is nothing else is there in the whole system. And you can pull it out and put a vehicle on which nanolipid you want to deliver. So this gives the advantage of two types. One is, this is the simplest possible technology, but what the viruses are mutating, you can immediately go back, and within 2 weeks, you can come back with a new vaccine. And you don't have to change anything. This is the first advantage. The second advantage I will talk later, where I will like to say, this is disease agnostic platform. So whether you want to make for H1N1, or you want to make for COVID, you want to make for tuberculosis, or you want to make from HPV, the whole facility is same. Every system is same. Only you have to change the DNA template. And this is why now people are saying this is going to be future of vaccinology. Now, when we decided to do this, this is, as I said to you, we said we will take the most prominent form at that time, which was the D614G. And we use one of the technological challenges, which is self-amplifying. Self-amplifying, you can make mRNA in such a fashion, which is one just like Moderna and Pfizer using single. But here, if you put a machinery against it, you can make multiple copies, so you have to give the low doses. And we also work on the chemistry part with one of our collaborators back in Seattle, that we will not entrap this. And that was there was a logic behind it. Why not to entrap, which is the standard technology available in the world. The biggest challenge in front of us was, can we make this vaccine 2 to 8 degrees stable? This was the real innovation behind it, because -70 infrastructure making the country will be very, very difficult all over the country. It will need Hercules tasks. So we said, can we make and can we innovate? And then can we use for mass vaccination and also the booster dose, the way we can go. And then we proved the proof of concept. The phase 1 trial was done, the data was analyzed, it was very positive. The government LSD funded 100 crores to scale up and phase 3 clinical trial. Now, why this thermostable vaccine, from where the inspiration came? And I would like to show you video. I do not know whether you can hear it. If not here, then
probably the first approved vaccines for COVID-19 will be mRNA, and yet we can't count on them alone, because they're very hard to scale that manufacturing, and they have a logistical problem because they require an ultra cold chain. Now, scaling these for the world would mean having hundreds of millions of doses in liquid nitrogen and keeping them at that negative 80 degrees Celsius as they're carried from plane out to get delivered in the world. I'm hopeful that as the mRNA platform matures in the years ahead, we'll solve both the scaling problems, which will bring the cost down, and will make this cold chain requirement unnecessary.
So this was presented, and in my first interaction with the Honorable Prime Minister, he said, "Do you think it is possible?" I said, "Scientifically, most of the things are possible, but I assure you, if anybody else will be able to solve it, we will be one of them." So now I'm going back chemistry into it. So both the vaccine which has gone into market, they are the design B. So this is nanolipid, and inside it there are small packets are there, and where mRNA is basically filled. So what looks like that in these small pockets, if you take a bigger view, you can see that inside also these pockets, water is there. And basically water is responsible for degradation of mRNA. So this is simple chemistry. So all our information is stored in form of DNA. And DNA gives the message to mRNA, mRNA makes the protein, and protein do the job. But somehow, because in our evolution, the evolution biology might have decided that we will keep the information in form of DNA. mRNA could be digested very quickly in a couple of hours. So this is the way they have put it together. And this all is because of one basically atom. And what happens if you go into the chemistry, and Professor Maselkar is there, he will appreciate very simple chemistry. This is hydroxyl group, and this is phosphodiester bond. And what happened? In presence of water, it just goes and it breaks it down, and the bases are broken down. So the major reason how we can take out the water from it. So in our case, the DNA is wrapped on the surface. And already this was coming up that few of the batches of Pfizer-BioNTech vaccine were near miss because they were having the water and it is continuously degrading it, even if you keep at minus 70 degrees centigrade. So potency was going down. So this was coming in on way. So basically we decided we took out the water by lyophilization, because our DNA is not put into packets inside the nanolipid. It is wrapped through a charges basis on the surface of the molecule, on the surface of nanolipid, and we have taken out the water, and this is thermostable. When we tested with minus 80 frozen liquid and lyophilized form, we found there is no difference, including its neutralization titers. So if you can see on the right side, the average range of conversion individual titers are between 40 to 60%, and both the vaccine gave around 80% plus. So basically what this knowledge of the bit of chemistry, knowledge bit of biology, knowledge bit of our production capabilities, we first attempted to try this thing. And today what happened, this vaccine is even stable for 60 days at 25 degrees centigrade. So this was amazing thing. This was a challenge for us, and we have able to solve the challenge. Why I'm presenting this particular part? We have to innovate the technologies deployable or needed to us. We don't only have to always follow the West. West has their different needs. We have their different needs. So going forward, these are the two big patents we filed. First, we filed that how RNA can be absorbed on the nanolipid emulsion on this. And the second one, we filed this patent on the RNA absorbed on the surface. So now I'm moving for the next part, and why I say defense and health Yes, we have Rafale, but we also need continuous missile supply if it has to fight. It is not that we have Rafale now, we don't have the backward integration and supplies are limited. Then, so it is very, very important for us. We have to do technology leapfrogging, but also back to back, we have to do self-reliance through backward integration. It is very important. We have to produce most of the things by our own. Certainly, we cannot produce all the things by our own, but we don't need to be totally dependent on outside for our import to if we are leapfrogging the technologies. I'm telling this word, if we are leapfrogging technology, we have to also seriously think about what is the self-reliance to backward integration. So here I want to talk about the technology. So Moderna and Pfizer and CureVac both gone on the simple mRNA, while Gennova we tried to do self-amplifying mRNA because we have to do the advantage of dose sparing. We don't have our clinical trial phase 2, phase 3 are 10 microgram, while Moderna is 100 microgram, Pfizer is 30 microgram. And this is what chemistry we talk about. We put the mRNA on the surface of through the charged lipid, and this is the way we have. There's a huge ease of manufacturability because people who are involved in this manufacturing of nanolipid, you have to entrap your mRNA and you have to put through a lot of pressures, a lot of losses happens. Here losses are hardly 5%, but we have gone into the physics dynamics and where we can just, under typical conditions, we have to mix through a specific instrument and by which it automatically mixes and losses are not more than 5%. The third differentiation in technology we innovated is lyophilization. And fourth, we are at present doing intradermal. And if this works out, then the most important thing, it is painless, needleless injection, and it may be highly effective because intradermal, when we are delivering the mRNA, it is directly getting loaded into the lymphatic system and responses looks like very, very high. So I'm saying we have gone into 4 parts of this technology independently. And once we have done 4 parts of this technology, now we are integrating it together and we are moving forward. Now, this is very important slide. And what I said, disease agnostic platform. And this is for the future of health security or healthcare, you can say. So we have this nanolipids, certain enzymes are required, certain cofactors required, nucleotides, lipids are required. We can create the as a stockpile, national stockpile. And once we have this national stockpile, if we have taken a plasmid, any virus, we have taken a plasmid and we have brought it here and we have added these things and finally the vaccine is ready. So how to integrate this? This is not only for COVID, but this is going to be a platform of this is what we are saying, there's a future of technology. And another part is if required within 54 days, once the platform is approved, once the play vaccine has gone to the market, within 50 days, we will have the vaccine ready for any new variant. So this is another example. The only thing what happens that many times our regulatory systems are not ready to embrace the new technologies. That part our policymaker has to think. Yes, they are often very helpful. They go into these kind of things, but somehow we have to bring the knowledge at the policy level where the regulations are there, how we can add the value into regulations. So I know the Professor Maselkar committee was there, which somehow get rid of ourselves from the environmental committee into the RCGM, but still RCGM need a lot of modifications because they do not have experience of taking the product to the market. They just go by science, certain guidelines, and sometimes the dialogues doesn't become very helpful. And those dialogues need to happen. When the Moderna and Pfizer vaccine has gone, EMEA and FDA people were sitting in their companies and helping them what is the best way, ensuring the safety and all these kind of things. So somehow we need to bring resurgence one more level at the policy level and regulatory level if we are leapfrogging the technology. This is very, very important. So now I'm just telling another part, which is self-reliance. So backward integration, none of these enzymes are available. So we have to buy these enzymes from all enzymes from outside, but now we have put our own program. We have made most of them, and now we are moving towards the GMP side. So this will not only reduce the our supply chain, but it will also going to reduce huge amount of cost for us for our vaccine. If we see, this is the whole program I have given like 39 or 40 raw materials, but there are raw materials which is colored. These are very, very critical. So we said, okay, go ahead, we will make it all by our own. So we have most of them, which are the critical one, we have already started the backward integration program, and within 6 months, we will be not only self-reliant for our supplies, but we may be also in position to export these things outside. So this is the technology part, has to leapfrogging technology part, has to be collaborated with Selfreliance. Otherwise, we will make it, but we will not able to use it. So with these words, I think I would like to thanks, and I'm open for discussion, and thank you very much.
Thank you, Dr. Sanjay Singh, for an excellent presentation. It was really very good. You covered right from the basic of what a vaccine is up to the levels at which the imports and exports would affect the synergy in our country. So thanks a lot. We will move on to the next talk immediately, and after that we will take the question and answers. Before we go to the next talk, I would just like to tell all the participants that there is a Q&A box down there. So if you have questions, kindly start putting it in this box so that we are able to take them at the end of the session. Thank you. And over to Dr. Mishra.
Thank you, Dr. Kale, for the introduction. I hope you can see the slides.
Yes, yes.
Yeah, okay, great. So first I would like to thank Dr. Mashelkar, Lieutenant General Patankar, Dr. Kale for creating this platform and inviting all of us to come together and discuss this. I would also like to thank Mr. Kotwal, who is not around, but we had several rounds of discussion with him to organize how to present this. Particularly, Dr. Mashelkar has been source of inspiration, and it's really such a learning for many of us. You just heard Dr. Sanjay Singh speak about his efforts in vaccine. He's very modest man. Several things he has not said, that he spoke about the technology and how useful it is. What he has done requires not only the technology and science, it requires creativity, courage, and really the desire to do something important for the country. And actually, my talk will be largely redundant because the spirit that he has put is what I was going to put across. So I'm going to describe a little bit during this pandemic what we learned and what were the positives that we thought came from this very difficult time, especially in the context of healthcare. And today we will put in the context of national security. This pandemic was really testing times for the healthcare system. We have seen this happening in front of us, huge economic impact and social and psychological impact we are still counting. So a country, even whole world is shaken up by one simple infectious disease. So when we are talking about national security, we have to keep this in mind. It's not only that, in fact, the increasing scope of the healthcare is going to be the major driver of economy and job creation. And that in fact is an important component for the national security. And as you just saw the glimpse from Dr. Singh's talk, the new era of healthcare is emerging, which involves precision and personalized medicine. We are not going to take drugs, everybody gets the same drug. We are going to have individualized thing when the genome comes more into the play. We are going to focus on prevention and nutrition and One Health, which means environment and animal health. They are going to be very important. And if we want to be a powerful and independent nation, we have to focus on these issues. Otherwise, we'll be left behind. And we have felt this in recent past during the pandemic, at how we were caught unprepared. When new infection or a pandemic arrives, whether it's natural or enemy intended, what do we need to do immediately? We need detection and diagnosis, which has to be accessible, which has to be feasible, high throughput methods have to be of this kind. We have to control the spread then, and there are many interventions. They may be technical, behavioral, governmental interventions, and there will be specific associated issues like new normals, what we call the new policies, and information and ideas have to come because many things are unpredictable. One more important thing which plays a very important role is communicating the unknown, and we have to be prepared for that. Keeping politicians, policymakers, academic peers, media, and general public informed, extremely important. And in times of uncertainty, it becomes extremely important for the people to have credible sources of information. Scientists and academicians acquire a specific role during this time, because it's extremely important because they are the knowledgeable for this. One more thing which is very tricky is that things keep changing, new information keeps coming. So it becomes very important that we keep discussing with the stakeholders like media, policymaker, and public, and adopt the best available at that time. What I'm going to discuss now are the multiple roles that CCMB has played during the fight against the COVID pandemic. And I would like to say two things. One, that CCMB is a small lab of CSIR. It's not a medical institution or IIT. So we had to really adapt very differently to get involved in this fight. And all I'm going to talk about is almost completely the contribution of our students. So I have the only role to just talk about these things. They are the ones who have driven all this, what I'm going to discuss in next few slides. So first thing CCMB did was to reorganize our labs and become a testing center, training center, validation center, and the national repository for the virus and patient samples for the SARS-CoV-2 in very quick time. We started culturing the virus so that we can use them for the testing and screening of drugs, for testing and validation of devices. And we developed diagnostic methods, protocols, SOPs. We address the biology of the virus to understand more about it and the genome dynamics of how the virus is spreading. So I will just give in next few minutes a few examples of what we, for example, what we did in diagnostic. This is all really now an inspiration that how our students respond. They are bright minds, and when they face a problem, they come up with a solution. So this is a common way of testing that was available at that time. So you take a swab, you put in some liquid, it's called viral transfer medium, and then make RNA, then convert into DNA and do the RT-PCR. It's a quite a demanding test. So what our student did was very simple change, that don't put in the liquid, just transport it dry. So there is no transport issue now, no leakage. You don't need to isolate RNA from this, and you can do test which is 3-4-fold higher throughput, less cost, less time, without any new science, just innovation. And of course, this was very well appreciated by industry. It was approved by ICMR, and many companies have taken license and they are manufacturing and even providing it for the test today. We joined hand with another company, Bangalore-based company, Singh, which is a Biocon subsidiary, to find a way of doing large-scale diagnostic. So I will not go into detail, but this technology allows you to do 50,000 samples in one go, and the cost will be something like 200 per test. And this you can do 50,000 in about 48 hours. So when it is required that the whole army unit has to be tested, or whole industrial complex has to be tested, this can be done in one go. Or when really big disasters come, you can do large amount of testing using this approach. It has, of course, demands of logistics to collect so many samples and put them in place, but technology is standardized and is there. We also, since we were culturing the virus, we started testing drugs and devices, and the huge response. I mean, we really flooded with the request. There is a private company, collaborations, various organizations. So far, we have tested more than 200 different drugs and compounds, and many of them have gone quite far in terms of their utility and applications. And I'll give you just 1 or 2 examples of this. So this was the very first drug that we tested with DRDO. DRDO approached us because we were culturing the virus. And as all of you know, Science Minister were able to give this to Defense Minister as a endogenous repurposed drug for this purpose. We had in our incubation center many entrepreneurs, and they were also involved in developing new ways of fighting this virus, and many of them are now commercialized. And this is the interaction with the company, Hyderabad-based company. They were making antibodies against snake bites and so on. So they came as we made the virus for them, which they injected in horses, purified antibodies. This was for therapeutic purpose, and this antibody is doing really well. So this will be actually an antibody therapy. It's now in a stage phase 1 and 2 trial, and doing very well. And I think this will be in the market sometime soon. So these were the requirements of that time from various industries and institutions. On our own, we were collecting the virus because we were the diagnostic lab. We were sequencing the virus in large scale, and we developed a platform to analyze this virus and find out what changes are happening and which direction the virus is going. It's crucial to track the emerging mutants by sequencing viral samples from patients. And any of you can go and just play with this app available on the net, and you can see how many genomes have been sequenced from India, how many mutations have been cataloged, how many lineages are that being tracked, and how many states are covered. And like this is, for example, the Delta, which is the most abundant at this moment. And we also do constant monitoring of what is happening till very, it almost real time in terms of data. So these are different variants that they are going around based on our sequencing. So we compile from the data all over the country and then monitor it here. So you can see that this yellow thing is actually the Delta. And some of you may have heard this AY 4.2 is this dark blue. And we are monitoring how they are doing and linking if there is anything to worry about these sequences, inform the authorities as and when required. And this being constantly done using our facilities. I will just digress for really a minute, that which I think is very, very important, and all of us should spread this message that there are some key factors that are variant independent. We really don't have to worry about the variants, which is to wear mask. Even if it is repetitive, we have to keep this in our mind. To defeat coronavirus, we have to use the mask, and not wearing mask in public is uncivil, and we should emphasize this point, because this one way we will be actually that will make the difference. And we have to avoid clustering, and we have to take the vaccines because they are the best means that are available for us now. Variants will emerge if we allow them to emerge. If we use these methods, then virus will be suffocated and it will not be coming with the new surprises. That's the purpose of this. During this, our students also interacted with neighboring labs, and they started collecting samples from sewage treatment plants and identify the pathogens from them. And it turns out that sewage or wastewater is a very reliable source for correct estimation of the number of cases present and even the variants. We can now even sequence the samples from the sewage treatment plants and tell which variant in what proportion is going around in the city. So this is going to be extremely important means of monitoring At the moment, of course, we are doing for COVID, but for many other diseases, we are developing this approach. And we have been doing in many cities, 10 cities at that time, and we were able to get very, very accurate number of what is the number. Imagine you don't have to go and collect any sample, just collect once in a day of appropriate time, and then you know what % of people are infected. Something which I think all the cities have to put in place. Now we are having a research grant from Rockefeller Foundation in which multiple centers are involved, Pune, Bangalore, Delhi, Hyderabad, and we will include more cities where we are doing close monitoring of this. There were a lot of uncertainties how the virus is spreading. So our students went to hospitals in different conditions, in COVID wards, ICUs, undressing rooms, OP corridors, and even we went to apartments of some volunteers where infected people were isolating themselves, and we collected the air from those places and checked for the virus, and we were able to establish that we can sample and detect the virus from the air, and we can even culture the virus that was collected in this kind of situation. And we even provided an advisory to the government and to many other organizations that what precaution needs to be taken. If we wear masks, even in the same room, we saw that people, the virus is safe. Public toilets were one of the key places where people go and try to remove. We saw that in the toilets for more than 2 hours, the virus is still hanging around because of the flushing, the aerosol is formed and is generally small and closed room. So if we are using public toilet, even if 2 hours before somebody has come and used, it might be still infectious, and we must not remove the mask there. One more component which Dr. Singh mentioned, that most of the pandemics or the new infection, they come from animals, and there is a reason for that, because closely related animals are having their own pathogens, and if we don't take care or we intervene or take the access where the wildlife is, where we shouldn't be doing, we are also exposing ourselves to the new pathogens. So these are called the Genocides, and these are the infections transmitted from animals to humans, and they can become transmissible from human to human, as is the case of SARS-CoV-2, and become a problem. So current pandemic is thought to have originated from the bats and come to a wet market in Wuhan city, and from where now it became human to human. We have been testing a lot of animals from the zoo. We've been getting requests, so all over the world of the many mammals which have been tested, 13 species can actually be infected by this virus, and some of them can even transmit. And we regularly monitor. So we had some cases in the Hyderabad Zoo. In the lion, some were infected, and we figured out how to avoid such a thing. How to, for example, how to make the meat that is given to them virus-free. You cannot spray a disinfectant on them. So just hot water was good enough. So those are the suggestions, simple suggestions that you have to give. This is important thing that wildlife is something which we are being very unfair to. So whether it's wildlife trade, whether it's wildlife consumption, or whether destruction of the forest, these are the things which are exposing us, and the potential spill from the wild is becoming more and more possible now. Human race must accept that this planet belongs to all the creatures. We are not the only owners that we can go and do whatever we feel like. This we have to learn and respect. Otherwise, we will be in big trouble in many different ways. And pandemic teaches us that it's time to take note of it. I will, next few minutes, I will spend on indigenization and self-sufficiency, how it is so important. We just heard that vaccine components we have to import. Similarly, for most of the things, whether it's supply of kits, medical devices, and a new generation of drug, we are really heavily dependent on import for every single thing. I mean, those who do research, they know that even good quality water for our experiment, we have to import. That is kind of not acceptable. So new generation of drugs and vaccines are going to be really the future of healthcare system, where there's antibody therapeutics, biotherapeutics, RNA therapy, mRNA vaccines. You just heard a fantastic display of how this is going to be a game changer. What Dr. Singh did not mention was that it's not only for the vaccine. You can use this technology even for cancer, for many other drugs, for genetic diseases, genetic disorders, because you are actually just providing the cells some message, and then the cells will use this message and provide whatever is the protein or signal needed for the body. And as I mentioned in the beginning, precision and personalized medicine. So we have to do very large scale genome sequencing at a population scale. So we need to be self-sufficient for that. Otherwise, we will be doing what powerful countries will want us to do or allow us to do. And genome and genome editing technology is going to be crucial in disease, nutrition, and environment. Many of us who have been closely involved in the response to SARS-CoV-2 or COVID-19 infection, this pandemic has given clear message that the basics of healthcare, India must be completely self-sufficient. We have seen that even simple reagents that we depend on import were in short supply. We had to wait for days and days and weeks. Something will come in Delhi and they say that one component is going to Chandigarh, one is going to Hyderabad, and we have to wait for one more week. It was really humiliating when we realized that we depend so much on these things. And please remember that when other nations who are the suppliers of these things, they will need for their people, we will have to wait. So country of this size, resource and capabilities must not be in such a situation ever. And there are many, many examples where we are just convinced that this is just not acceptable. Question is, can it be done? So you just heard Dr. Sanjay Singh showing that it can be done. So of course, yes, we can. And I'm going to give you just one example of this before I stop, which was, as you know, that most components of the diagnostic kits were imported. So with some organizations, we decided to indigenize it completely. And we made a plan. This was done really like a professional company doing it, not a relaxed research institution doing. So we clearly defined our objective that we want the testing kit to have everything indigenous, all the way from sourcing of the components to the marketing. And we defined all these steps. You don't have to go through this. It's just to impress you that there was a monthly plan, even more detailed plan of this. And each component was considered. We will get from where, how to indigenize, who will be the company, who will make it internally. And here is the result, that in about 8-9 months, we could reach a point where everything can be indigenously made and the cost of the test will be 68 rupees per test. Of course, it doesn't include human resource and margins. And so this is now doable. If you remember, in the beginning of pandemic, we were paying 3,000, 4,000 rupees per test. We can do now in 100 rupees. We've identified companies who with GMP facility who will do packaging, identified the companies who will be marketing. Today, the production capacity is 0.5 million reaction per month, and it can be enhanced to 3 million by organizing and sourcing. This can be done. So yes, we can. When we want, we can do it. And it is time that we take note of it. In future pandemic situations, as we have just heard, that pandemics will keep coming and we have to be prepared for that. So our surveillance has to be in place, whether it's for vectors like mosquitoes and flies or parasites. It can be virus, protozoan and bacteria. Zoonotic disease surveillance is extremely important, particularly where we are interacting with the wildlife, because there are increasing wildlife people contacts that has to be controlled. Cattle people contact and consumption of wildlife, these are the factors that are important in this. And we need preparedness for the quick diagnostics, adaptation, and indigenization, as I have been saying again and again. So in surveillance, there are, I just gave a small example of the sewage surveillance. There are genome-based surveillance is now possible. We are sequencing thousands and thousands of patient samples for the viral genome, and we have pretty good idea of how the variants are emerging and which direction the virus has been going, and for what are the sources and so on. And that has to be in the place, so that next time when it comes, we don't have to make a new INSACOG. There should be permanent system to handle such things. And we should be prepared for existing infection for which we know. And also, as you heard, there are new infections that will come because they are there in a reservoir in the wild. They will come when we interfere there or by some other accident and so on. And antibiotic resistance is another important thing which can create as a problem. We are talking about natural pandemics. It can also be a situation of bioterrorism. Yesterday, you heard Mr. Dole pointing out that threats are of various kinds that we have. And bioterrorism is one thing which we should keep in mind, because these are going to be the weapons in the future that may be used. So they can be released in the air, they can be released in the water, or by other means that has happened earlier for the anthrax. And if they're infectious, then the affected individuals become the vectors and they start spreading unless we identify, isolate, and treat them. Exactly the case of current pandemic. So the combating the bioterrorism threat requires preparedness of this triangle of detection, containment, and treatment. We have to have things in place for that, and methods for rapid and effective very low concentration detection is already there. Is a DNA-based microorganism identification, and this can be done. This is such a sensitive method, even few molecules, few pathogens can be detected. So we can do it. So let's do it. And I think we have learned enough lessons from this pandemic. And the system has realized that there is a capacity, but we need to pay attention, and we should not be again caught unprepared for any natural or enemy intended situation that can emerge in this way. So I'd like to just thank you for your attention and giving me an opportunity to talk. And as I said, a special thanks to the student community of CCMB. All the work I talked about is done by them, thought by them, and so on. So thank you very much, and I'll be happy to answer if there are any questions.
Thank you. Thanks a lot, Dr. Mishra, for a very, very good talk. I especially could note that every pandemic or every virus need not be a very bad situation to be going into when we consider the way in which the institute has shown the preparedness in these current times. The way in which the students you have in CCMB have been graduated to become not only a research student, but also to do the testing, do the training, validation, and creating a repository, and then also supporting the entrepreneurs for the kind of kits that they have been developing. Also the way in which you have done the air sampling, the sewage sampling, which will go to the environmental level, and how to have the hygiene, personal hygiene, as well as the way in which animals have to be protected, and the way in which bioterrorism can be also managed and handled. Thanks a lot for covering a very wide range of canvas of this particular topic, especially from the perspective of an institute or a student-centric community. Thanks a lot.
Thank you.
We have actually many questions in the Q&A box. So we'll take these questions first, and then I would request the other panelists if they have specific comments or any questions to be asked. So let me go to some questions. Some of them are answered, but anyway, I would like to put them here also on this platform so that everybody knows about it. So there is a question from Vinod Menon, and he said, sorry, that's the question for the yesterday's session. Sorry about that. The next question is by Jyotishwar. He's asking that asking this question to Dr. Sanjay. My question is that although clinical trials and early studies of these vaccines in real world use have shown largely positive results, any thoughts on long-term effects which are still unknown? Dr. Sanjay.
So that's absolutely right. At present, both the vaccine has been given to close to 200 million people worldwide, and what the data latest shows from CDC, these are the safest of the vaccines and also most effective. Long-term effect, now almost 8 months, 9 months has passed. We do not see any systemic event because mRNA doesn't enter into the cell. mRNA doesn't need to enter the cell, it remains outside, it doesn't hit the nucleus basically inside. So if it doesn't need hit nucleus, there is no chance of this going to be integrated with our genome system. So this is why mRNA is treated as a much safer and much effective way to do the vaccination. Yes, DNA should also be safe, adenovirus also should be safe, but theoretically and scientifically, mRNA is super safe because it doesn't integrate with the genome system. Now, for the vaccines, when they are launched, the side effect or long-term effect, it takes 5 to 10 years to evaluate. But till now, we don't see any major issues as per CDC surveillance system, which has above 150 million doses and very effective surveillance system.
Okay, thank you, sir. The next question is, what would be the impact of having robust mRNA vaccine national ecosystem in reducing our national risk towards bioterrorism?
This is a very important point, and that is why I said some national stockpile. So at present in US, the BARDA is already talking to companies, and they have even reached to us that whether we can stockpile these things on their price and keep in store. The same way, once the vaccine goes to the market, we will be in deep discussion with the government on these points, because it looks like mRNA is a technology which can within no time you can respond it back, and these are the bioterrorism, most of the viral infections. So that will be an absolute answer as we see it, but certainly it is going to be the part of the national stockpile.
Thank you. Next question is for Dr. Mishra. Would CSIR be able to drive a national mission on indigenization proactively when there is no emergency? We have reacted well in the case of emergency situations. So the question is, would CSIR be able to drive the national mission for indigenization?
Yeah, actually, I had focused this talk, but CSIR is already involved in this. We used to have, you will not believe, every day meeting 6 to 8 with the DG, every day meeting with our core group and discussing what is happening. Large number of initiatives have been already put in place in the leadership of Dr. Mande, which involves PPE, sanitizers, now air purifiers, so many things. In new parliament building, we are going to install air purifier, which will make the air viral, safe, and so on. And CSIR is very powerful organization in terms of scientific capacity. There's 38 laboratories across the country. So they are strategically very well placed. So certainly this can be done. There is no question. I gave a small example of something which was spontaneous kind of response of the student community, and of course then industry interest and so on. When we plan at large scale, it can certainly be done. These are all, many of these are not very high-tech things. What we need to do is to do quality control and manufacturing. These are the two things that, and institutions like CCMB or a national institution, they can be just doing the quality control. Industry is very, very capable. I mean, we are making all sophisticated things. So I think now we have sensed the taste of success, though the hard way, but I think we will not let it go waste and turn the table and put in our favor. And this can be done, sure.
Absolutely, absolutely. Next question is by Mr. Atul Joshi. Long COVID is a term that has emerged in this pandemic. It has been a cause of vaccine reluctance or hesitancy. Can the panel throw some light on it?
So this is little bit, I will redefine the question. So long COVID is used at present as a term that when people get infected and with comorbidity, they are not recovering well even after months and months of treatment. So that is basically the long COVID term which has been used. But I think the question which has been asked, that what is the future of this pandemic? The only one future of this pandemic, we have to immunize effectively more than 80% of the population. There is no other answer. Because many diseases we have eradicated only on the power of vaccination. So the government is putting its best effort, and I hope that the drive is going very well. We will be able to control in this. And then we also should be ready maybe for the booster dose where the chances it can come back. Because what has happened in either you talk about Israel or talk about US where they have tried to do it, some part has been contributed by vaccine hesitancy and some part has been came back because of the age-affected poor immune response. So we have to control this virus spread as much as possible. The last thing I would like to say, the real control would be that we should have very high antenna in terms of surveillance. If the new virus emerges anywhere, we have to contain that into that localized area, and we have to understand, and if needed, revaccination has to be done. This is the only way we can totally eradicate this disease. And I'm very positive we will eradicate, we have eradicated polio.
Thanks a lot. Very nice and a very, very positive answer. Any other questions, any comments from the panelist members?
If I
Yes,
can,
sir. Yes, sir.
I mean, yeah. First of all, a fantastic session. Very, very proud. I said we have got 2 stars. Of course, we have 3, including you, Sangeeta. But you make us very proud. You know, it shows, like, I loved your slides when you say, yes, we can, yes, we can, yes, we can. I think this is a fantastic demonstration of what we can do. I always talk about talent, technology, and trust. We have all the talent in the world. We have the technology, as you have seen. It's a question of trust, believing in ourselves, all right? And I think that is the crux of the matter, to be honest. There are 2-3 specific questions that came up, so I will just add to that. Sanjay, you mentioned about the regulatory apparatus and how that has to match the pace with the developments that are taking place in technology. And I think that's a very important point. I remember, and you mentioned about the Mashilkar Committee, 2003. And since you mentioned Mashilkar, I have to say something about it. 2003, under my chairmanship, there was a committee to actually look at reforming the entire drug regulatory system. There is called a 2003 Mashilkar Committee report. There have been 16 Mashilkar Committee reports, but 2003, this report. And I remember on a lighter vein, these Priyadarshini International Awards were being given, of which I was the chairman, and Ghulam Nabi Abad, the health minister, was the chief guest. And in his remarks, he said, in our health ministry, we remember Mashalkar every day. So Kumar Mukula Birla, who was also awarded, he was sitting next to me. He said, "Why do they love you so much?" I said, "That is because industry is going to them practically every day and say, implement Mashalkar committee report." So what has happened is that that report was absolutely far-reaching in terms of complete reconfiguration and making it really a scientific organization. Unfortunately, it has been partly implemented, but not fully. That is the issue. I think we need to Rakesh, you will remember you talked about the dry swab. Okay, a fantastic technology. But your DG, Shekhar Mande, he participated in our Science and Society program 7 or 8 months ago. And he was openly critical of the fact that that technology was not getting cleared. And you have gone through enormous issues. You were nice in not sort of disclosing that, but the technology that benefits this thing. And there were intricate factors why it was being held up. So who is our worst enemy? We.
Ourselves. Yeah.
Right. I think we need to really change our mindsets, to be honest. Our scientists are fantastic, technology are fantastic. It's a question of trust, Sangeeta. You know, I'll just take a minute. You know, for example, and both Sanjay and Rakesh will know, you know, the Anjani Masika Inclusive Innovation Award, one of them went for this dengue test, you know, that I see there we had sort of created Dr. Naveen Khanna. You know, he did something where it could be detected within 15 minutes as against 1 or 2 days. But we are sort of set on importing them from South Korea, from Australia, and from US. And when the pandemic broke up, actually, when everything was getting exhausted, the only country who said we'll give it will be South Korea. And South Korea loaded those diagnostic kits on the wrong ship and it went to Africa. So we didn't have any. And then they started using Naveen Khanna's, and today his market share is 80%. Okay, but if that ship had come, basically it would be still using South Korea. And so I think as an old man, I'm sorry, I feel a little emotional because I see this fantastic talent, and what is holding us back is our own trust, confidence. And I think your two spectacular presentations have given us that trust and sort of confidence. I would also say that Sanjay, you talked about a very important point about we have to do things for India, for Indian conditions. And this is true, eternal truth, as a matter of fact. And in this particular case, you are having it. So this is not just innovation, Indian innovation for our conditions. So as to say, we can't have minus 80 degrees centigrade. We have 5 degrees centigrade is wanted. And the last point I'd like to make is that is why all the more, you see, Sanjay, when you talk about mRNA and within a year, people coming out, you know, Moderna and all that, etc. But don't forget that in mRNA, in the science itself, there were so many years of efforts that were involved. If that was not there, you could not have come. And therefore, investment in science is very critical. And I'm sorry that in 1998, 0.68% of GDP was our R&D expenditure. In 2021, it is 0.68%. You know, and there have been good intentions to raise it to 2%, but I think these two presentations show as to why our investments in science have to go up from what they are, and very substantial. These are 3 or 4 points I thought I would.
So I would like to answer, and this is fine because this is what you said very close to my heart. So first, Sangeeta, I'm also the product of CSIR. I did my PhD at one of the CSIR institutions. Very proud, and Professor Dr. Maselkar is always will be praised. Sir, I don't hold my guns with the regulators. Even Dr. Mishra knows. If the policies are influenced by not knowing the knowledge with biased intention, we have to fight. We have to stand up against it, and we have to openly challenge it. This is the only way we can change the system. Openly, we have to challenge the system. One of our products for a stroke, which is today is one of the highest sellers in the country, We went for 9 years for regulatory approval. But it didn't stop. We fought and fought and fought. We got the approval, and it was challenged in the court next day by one of our doctors in the country who put a PIL. We went and fought in the court with the top 2 lawyers and beaten them down. If we believe in our science, we should not hold back. We must fight with the system, we must fight with the regulator, and we must teach the lesson to the policymakers. Because most of the policymakers, sometime I found somebody asked, what is the cost of the vaccine? And I said, the cost of vaccine is how to stop this pandemic. It costed more than $1 trillion to Indian economy. So vaccines should be safe and effective, which can stop the pandemic. That is the cost of the vaccine. So sometimes we have to bring these new thoughts together with the old thoughts, because I have said personally to the government, very openly to the top person, that we are worrying about 5 trillion economy. Microsoft, Google, Amazon, Apple, Tesla, combined is 10.5 trillion economy by 5 companies. The only way to raise our economy, raise our prosperity is through science and technology. And somehow it has to be taken on the highest pedal. And Dr. Maselkar, it has been heard very well. This is last week only. It has been heard very well. And I'm openly saying we have to promote, other areas are important, but we have to promote science and technology if I have to fight the battle. It is very difficult period which we have gone through, but only science and technology and the work like Dr. Mishra's lab and these labs has helped us to control it. So science and technology is very important. And my closing remark, especially for this national health security, science and technology is paramount.
Very good. All that I would say is that we can't be expected to run in sand and compete with Carl Lewis. So what we require, the .com that we require is a hassle-free.com. All right, that's all that one is talking about. You didn't have to sweat it out for 9 years for what was right and just. You get the point. We can't compete otherwise. I think that's the point that is being made. Okay, I think we are coming to 12 o'clock.
We are coming to the end of the session. Let me conclude the session. It has been an excellent session with a lot of good question answers, lot of good discussions, and extremely good thoughts put up by both the speakers, Dr. Sanjay Singh and Dr. Rakesh Mishra. Let me thank both the speakers. Let me thank all the panelists for a very lively discussion, and also, of course, all the audience who had asked many questions. Actually, there are many questions also still in the question answer box, but I would request them to connect with the speakers independently, because we do not have that much time to go through the questions. So let me thank the speakers again. Thank you, Mishra sir. Thank you, Vaidyaji, Abhay, and thanks a lot.
Thank you. In fact, it's been such a fascinating session. After last evening's session, where we were overwhelmed by the thought of those two kinds of threats. One was the biological threat and the other one was the cyber threat. Today, we are reassured that we are well on top of those threats. You know, I mean, that is such a happy feeling within oneself as human beings and as Indians, more as Indians than I think human beings. And allow me, because in fact, I'll use my prerogative as the convener to just say a few things just to put matters in perspective. You know, till the pandemic hit us last year, there were only one kind of warriors, the guys who were at the border, and they were being referred to as warriors. Then, since last year, and rightly so, everybody who's fighting the pandemic, the doctors, the nursing staff, anybody and everybody who was associated with this battle were being termed as warriors. We haven't recognized the third warriors, which are our scientists. I think today's presentation has brought that out loud and clear, that these are our warriors on an unknown front or an unseen front, which is probably more important than the front, which is the line of control or line of actual control or whatever, the seaboard on both the shores. Like last evening, I said that what we see as threats is only the tip of the iceberg. 7/8 of that threat is below the surface, and today we saw what is the scale of that threat, but how well prepared we are going to be. And both of you, sir, Dr. Sanjay Singh and Dr. Mishra, after what you said and that can-do spirit that came through so strongly, I think each one of us goes away and takes away that degree of confidence as citizens of this country that we can and we will. Thank you very much. Been a fascinating session. You also have given us tremendous amount of recommendations, and may I thank you by a round of hand clap.
Thank you so much.
Thank you, Sanjay. Thank you, Rakesh. Yes,
Yeah.
thank you. Thank you very much.
Thank you.
Our next and final session will be this evening at 6 o'clock. And as always, we'll be on the air from 5:30 onwards. So look forward to seeing you then.